Okay, so welcome to this next video in the playlist on calcium signaling. In this video, what we're going to do is look at skeletal muscle contraction, which is a physiological process to produce the contraction of a muscle. So skeletal muscle contraction. So this is the setup for this video. What we're going to do is begin by looking at skeletal muscle structure. Then we're going to look at the process of excitation-contraction coupling, i.e. how you change an electrical signal coming from a neuron into an actual contraction of the skeletal muscle, and then we'll look at the mechanisms by which the skeletal muscle actually does contract and shorten in length. And then finally, what we'll do is we'll look at the relaxation mechanisms. Right, okay, so let's begin by looking at skeletal muscle structure then. Okay, so let me draw a skeletal muscle here. So what I've done is I've basically taken a skeletal muscle and I've taken a cross-section through it. So it will go back like this maybe. Where should we begin this? Sort of like here. And it would end in a tendon maybe over there. So there's its tendon. So what I've done is I've just taken the muscle and cut it. Taken a cross-section through it, basically. And what you see is something that looks kind of like this. You see a sort of white connective tissue layer on the outside that surrounds the entire muscle, basically. And this connective tissue layer is what is known as the epimysium. So this layer of connective tissue surrounding the entire muscle is known as the epimysium. So this is the epimysium. Right, okay. Then, within the epimysium, what you see is loads and loads of these red structures. So, this epimysium would look white. It's white connective tissue. And now, within the actual, within the belly of the muscle inside, you have loads of these red structures. And then they are surrounded, this network around them is again white connective tissue. So this again would look white. And this connective tissue that surrounds each one of these actual contractile or red tissue here, that's known as the perimysium. Right, so I now need to tell you what is the name of these red structures. So they are known as fasciculi, or a singular is a fasciculus. So this is a fasciculus. Okay, so fasciculus. Right, and to get the point across, what I will do is I will colour them in red. So these are the actual muscles. Well, these are where the muscle fibres are, and they're what we're going to actually contract. And the rest of what they're suspended in is just connective tissue. So they're suspended in the perimysium, which is connecting the different fasciculi together. And then around the fasciculi, which are connected by this perimysium, you've then got the epimysium around that. Now let's pull out one of these fasciculi, or a fasciculus, and let's have a look at its structure. So now let's pull out a fasciculus, let's zoom in on it so it's now bigger. And basically what you find is that it's got a very similar structure to this largest muscle structure. So you find loads of little, again, contractile structures tightly packed within it. Okay, and these are the actual muscle fibers, the myofibers. And these are the equivalent of cells, but these aren't normal cells. These are absolute giants of cells. I mean, when you look down the light microscope for the first time at one of these are giants of cells. You can see them at low magnification. You don't need to zoom in to the highest magnification to actually see them. You see them at low magnification, and they are giants. And effectively what they are is they are loads and loads of cells that have fused together, basically, and formed a huge, great structure, a syncytium that's known. When loads of cells fuse together to make a larger cell, it's called a syncytium. Okay, so another example is the syncytiotrophoblast, which forms the placenta in embryology. Okay, so these circles that I've drawn in here, these are myofibers or muscle fibers, and I'll colour them in pink to denote that they are the bits that are actually going to contract. Now I won't colour in all of them because that will take time, but the name of these are, you can either refer to them as muscle fibers, spelt like this, fibers, don't write it the other way, fiber meaning like broccoli and things, things that we can't digest, muscle fibers, or you can denote them as myofibers. So myo means muscle, and then you just have a single word to denote them, basically. So muscle fibers or a myofiber, whatever you want to call it. And this connective tissue that surrounds the myofibers in a fasciculus, that is known as the endomysium. So the endomysium. Okay, right. Now, as I say, these muscle fibers or myofibers, they are single cells, but they are giants of cells. They have multiple nuclei for a start. They don't just have one nucleus, which is why we think they were fused from loads and loads of cells. Their structure is quite interesting as well. So if we pull out now one of these muscle fibers and look at its structure. So let's have one of these muscle fibers, and now I'm pulling it out and spreading it out longitudinally. So they are huge, great fibers like so. Okay, so this denotes our muscle fiber, or our myofiber. And basically what you find is if you look at the structure of one of these muscle fibers, or a myofiber, whatever you want to call it, basically it's got loads of little units within it. What you see is that it's divided up into loads of little strips, and these strips are all contracting on that. Well, they're all sort of like contractile strips, so I'll show it. Basically we will see later that the contractile unit of muscle fiber, what actually causes the contraction, is these structures known as sarcomeres. And basically the sarcomeres are a specific arrangement of the cytoskeleton that allows you to have contraction. So these are the bits that are actually going to contract. And you have another protein in between those. So like this. Okay, right. So actually I'll go over the structure of a sarcomere now, and then we'll see it later, because otherwise this picture isn't going to make any sense. Right, so what are these things that I'm drawing here? Well, basically what they are is they consist of a disc of protein. So here's a disc of protein within the cytoplasm, so a disc of protein, and this disc of protein is in the same sort of axis as if you took a cross-section through this myofiber. So basically the disc will be in the same sort of plane as this cross-section of the myofiber. So if I were to cut this myofiber down the center, then one of these discs would be in that sort of a plane, basically. Right, so you have these discs of protein, and these discs are known as Z discs, okay? And basically from these Z discs you have loads of other protein filaments attached. So I just want to colour in this Z disc. So I'm going to colour this Z disc in green, like so. Okay, so that's our disc of protein. And basically, from this Z disc, you have loads of other protein filaments attached. And I'm going to draw a protein filament here. And these protein filaments are basically polymerizations of a smaller protein. So these small little balls I'm drawing here, those are actin monomers. So each one of those little circles that I've drawn is an actin protein. So actin is a protein which is produced by the myofiber, and actin polymerizes together. So loads of these little actin monomers join together to make this larger fiber, basically. And this fiber that you've made out of actin is known as an actin filament. Okay, so basically from this Z disc, which is what I've drawn here that's the Z disc, you have loads of these protein filaments coming out from the Z disc, and those are the actin filaments. So rather than drawing each one of them in this way, where I draw each actin monomer bit by bit, I'm just going to instead denote the actin filaments by lines. Okay, so you have actin filaments coming out this way, but you also have actin filaments going out the other way from the Z disc, so like so. Okay, so the Z disc is basically this proteinaceous disc with with actin filaments basically spreading out along this way, in this direction along the neuron, and also in the opposite direction along the neuron. So that's what I've shown here. Here is a Z disc here, and here is its actin filaments going either way along the axis of the myofiber. Right, okay, so you have these structures in the muscle. And what you'll have is you'll have multiple of these. So you'll have another Z disc potentially over here. So here's another Z disc, and here are its actin filaments stretching back towards you. So here's another Z disc, and those are its actin filaments, and it's also got actin filaments obviously coming this other way over here. Right, okay, the structure that is within myofibers is basically lines and lines of these Z discs. So you'll also have more and more of these Z discs, and you'll have a whole line of Z discs like that. And basically that structure of loads and loads of Z discs all lined up is then known as a myofibril. Okay? And you'll have multiple myofibrils within your muscle fiber. So basically you'll have another row of these Z discs down here. Okay, right, so here's another Z disc, here's another Z disc, and that will basically make you up another myofibril. So here's a second myofibril. So you have multiple myofibrils, multiple of these lines of contractile units, and I haven't yet explained to you how they're contractile units, so I'll just finish off my explanation. So basically what you have is you have another different type of disc of protein here. So here we go. From which a different protein filament is attached. So let me explain the other protein that is essential in muscle contraction. So there's another protein, the structure of which looks slightly like this. So this protein is known as myosin. And basically, myosin has two distinct components. It has a fibrous tail and it has a globular head. And basically, myosin, again, just like actin, can polymerize, and it forms myosin filaments where the fibrous tails all line together to form this myosin filament, and then you have the heads of the myosin monomers sticking out from the myosin filament, like so. And your myosin filament, therefore, overall ends up as this strip of the fibrous tails of the myosin proteins all joined together with loads of myosin heads attached. And basically this is what is known as a myosin filament. So when myosin monomers polymerize together just like the actin monomers, it creates you a myosin filament. Okay, so we'll continue this discussion in the next video.